Hi everyone, my name is Annabelle Taylor and my topic for the PB480 term paper was about influencing secondary cell wall deposition through NAC transcription factor regulation of auxin. So first I want to go into just some basics about NAC transcription factors. NAC stands for NAM-ATAF1 CUC2 proteins. They are a large family of transcription factors found primarily in land plants. They have many functions, mostly in plant development, nutrient distribution, cell wall biosynthesis, and stress response. And also the upstream signal of the NAC master switches in fiber development is still relatively unknown, which is part of what my paper focused on. So this image over here on the right is transcription factor proteins and their basically their role in secondary cell wall development. You can see over here the structural genes of cellulose, hemicellulose, and lignin, which we know make up a lot of the cell walls. And so tier 1 is those proteins that are capable of binding directly to structural genes. Tier 2 is capable of regulating tier 1, and tier 3 is capable of both direct binding to structural genes as well as regulating tier 2. And you'll notice that a lot of these are NAC proteins actually. So SND 1, 2, and 3, as well as NST, BND, and XND are all NAC transcription factor proteins. So NAC clearly has a strong correlation with the secondary cell wall. So what about auxin? Obviously, we know auxin has many important roles in stem elongation, phototropism, apical dominance. So it only makes sense that these roles extend into auxin signaling pathways in secondary cell wall development. This image shows the significance of co-expression of auxin genes and secondary wall NAC proteins. This just elucidates how auxin genes are related to secondary wall NACs, as we can see some of the proteins from the last slide showing up here again, such as SND here, NST here, and VND. The paper that this image actually came from discussed how treatment of woody species with auxin resulted in reduced transcription of NAC transcription factors associated with cell wall development, essentially meaning that auxin treatment in the study resulted in thinner secondary cell walls. So to understand the primary article I chose, I need to explain a little bit about lateral organ boundary domain 29, or LBD29. It is found downstream of auxin response factors ARF7 and ARF19, and has been shown to have a role in lateral root formation in cell cycle progression in Arabidopsis. The image on the left shows the difference in lateral root formation between repressed LBD29 expression, wild type, and 35S LBD29, which is constitutively expressed. So clearly the 35S promoter and the constitutive expression of LBD29 cause much more lateral root growth. On the top right, the fluorescence indicates the expression of a GFP pin1 construct in leaf primordia, and just below that, the graph measures the expression levels of multiple pin proteins for these lines. Clearly, the high expression of LBD29 resulted in higher expression of pin proteins, which, just as a reminder, are proteins that help with transmembrane transport of auxin. So there is a strong association between LB29 and auxin movement. So why does this matter in the first place? Well, obviously secondary cell walls are incredibly strong. We all know this. They provide plants with the physical strength needed to grow vertically. They are protective, and they can constitute up to 90% of total cell wall thickness. The composition is mainly cellulose, lignin, pectin, hemicellulose. It conforms a matrix, and that is part of what makes it so strong. This also makes breakdown of plant materials very tough. By being able to manipulate the deposition of the secondary cell wall, plant products could advance in numerous ways, namely through strengthening of cell walls and fiber cells. You could increase the overall strength of plant fibers for things such as linens, papers, cordage. Same thing goes for plastic composites that are being reinforced with plant fiber. The weakening of cell walls is another option that could be better understood. To make cellulose and biomass available to enzymes, pre-treatment is often required, but this adds additional costs and has undesirable byproducts. So the deliberate weakening of cell walls to avoid large pretreatments could be one method of making a more cost-effective and environmental biofuel feedstock. But to do any of these applications, we first need to understand the many factors that affect the synthesis, deposition, and upkeep of cell walls. This presentation specifically is focusing on further understanding NAC transcription factors, auxin signaling, and LBD29. So this was my primary research article. LBD29 involved auxin signaling, represses NAC master regulators, and fiber wall biosynthesis. The hypothesis of this paper was that LBD29 operates upstream of NAC domain master regulators and is induced by auxin signaling. If LBD29 levels are increased, then secondary cell wall biosynthesis will be reduced and vice versa. So first, the authors discussed the identification of a mutant that they labeled FLSD, the fibrillus phenotype. As can be seen in the far left photo, the main phenotypic trait of this mutant was being extremely dwarf with very little vertical growth. You can see that this here is the wild type and this one is FLSD. There is basically no growth there. So the next thing they did was determine the LBD29 was the cause of this mutant, which they did by inserting an activation tag in the FLSD genome, followed by upregulation of LBD29 transcription. The graph here shows that the transgenic lines they created have a lot of this LBD29, and that the result in these cross sections you can see is that there was very little secondary cell wall staining. So here you can see the wild type has a very strong line, and here you can see it's very dispersed and almost not there. Next, they repressed LBD29 using a LBD29 SRDX construct, resulting in a noticeable uptick in the amount of secondary cell wall present, even more so than the wild type. So here you can see the wild type has a secondary cell wall, and then here the staining is even stronger when LBD29 is repressed. So at this point, we've learned about the fibrillus FLSD mutant, that this mutant is caused by LBD29 expression, indicating a correlation between LBD29 and decreased wall strength, and also that repression of LBD29 increases cell wall development. Next, using qPCR analysis of NST1, 2, and 3, which you'll remember are NAC transcription factors, the authors quantified how much expression of this NAC regulator was present in homozygous and heterozygous FLSD mutants. So this is the FLSD homozygous, this is FLSD heterozygous, and then over here they also have an LBD29 loss of function mutant, which they also analyzed the relative expression of NST1, 2, and 3. Both versions of the FLSD mutant had significantly less expression of these NST proteins, while the LBD29 loss of function mutants had increased NST expression. However, forced expression of NST1, 2, and 3 in FLSD mutants, seen in the bottom left image, resulted in suppression of the mutant phenotypes, especially NST1 and 2, which regained its height and secondary cell wall strength. So here you can see this is just FLSD mutant, so it doesn't have the secondary cell wall, but when you overexpress NST1 and 2, it returns, and you can see that this also correlates with the increase in height back to what I would assume is near the wild type. And finally, to prove that auxin is related to secondary cell wall repression, wild type and LBD29 loss of function mutants were treated with IAA. Wild type lines treated with auxin showed decreased secondary cell wall presence, whereas LBD29 loss of function mutants treated with auxin show very little effect, essentially meaning that while auxin has negative effects on cell wall thickening, it is reliant on auxin signaling involving LBD29. So it's time to put it all together. The most significant thing we could take from this research is that LBD29 is involved with inhibition of cell wall thickening due to the role it plays in auxin signaling. Also, the fibrillus FLSD phenotype that resulted in secondary cell wall deficiencies was caused by LBD29 and is responsible for regulation of NAC transcription factors. One thing I wish had been talked about more in the discussion, though, is that they mentioned how other studies had reported auxin as having both positive and negative roles for secondary cell walls, but it is not explained any further. They basically mention it, but then they don't say whether it does actually have positive and negative roles, or whether the studies are based off of just different discrepancies, whether it's the methods that differ in obtaining these results. So I wish they had gone a little bit more into why auxin would have both the positive and negative roles and what this could mean for their own study. Now, obviously, this is far from the end of secondary cell wall research, And so next, I think that there should be a focus on describing the effects that fluctuating auxin levels have on cell wall biosynthesis and regulation. Also, a more in-depth understanding of the pathway through which Ogden signals for cell wall repression would be beneficial for manipulation of its strength. But if you take away anything from my topic and presentation, I want it to be this. Manipulating the cell wall creates opportunities to enhance plant products like bioethanol, linens, and plastic composites. These enhancements are both structural and economic. LBD29 has important roles in auxin signaling that helps with the inhibition of secondary cell wall development. This is in addition to the other roles we know of, such as lateral root formation and cell cycle progression. High auxin levels result in negative repression of secondary cell wall deposition via auxin signaling, making it a negative repressor. And finally, LBD29 functions as an upstream regulator of the NAC transcription factor, which goes back to the hypothesis that I mentioned earlier. Here are my references, and thank you for watching. Have a great end of the semester, and hopefully everyone has great finals.